Hi guys, welcome to this part of SciFest about bacteria and antibiotics. I guess if you have clicked on the title, it's because you are interested to learning a little bit more about bacteria and the ways that we have to treat bacterial infections, which are antibiotics. So in this video, we are gonna give you a run through about our bacterial world, like it says here, about where you can find bacteria, where bacteria are normally living, where do antibiotics actually come from, and also a little bit about the real antibiotics that we can get from the doctor when we actually need them.
I think a lot of people haven't always thought about bacteria in the sense of where they are, what they are, all of that. So Ava, can you see bacteria with your bare eyes?
I wish, because that would mean I have really good eyesight. No, we cannot see bacteria with our bare eyes, but bacteria are actually everywhere.
Yes, they're on all surfaces, pretty much. They're on us, in us, in the ground outside, in the water outside.
Microscopes allow us to see the bacteria, the very, 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 very tiny bacteria, as single organisms. And this is what they look like.
Yeah.
We have here some photos of bacteria that are actually growing in different parts of the body. The photos that you are looking at right now, they are called micrographs because they are photos taken with microscopes, and they come from 3 different parts of the body. And what we want you to do right now, we are gonna put each one of the photos, we're gonna leave it for 5 seconds, and we want you to think about where in the body these bacteria are actually growing. Let's play this game, okay?
Yeah.
So, first photo. Jenny, do you know where these bacteria are coming from?
So this is actually from your mouth.
Yes.
Or a person's mouth, not yours specifically, of course.
Yes. No, my mouth. But what you can see here is actually a quite diverse environment with bacteria. Some are elongated, some are a little bit more like filament, some are pretty round. Then we're gonna do the second picture. So here you're gonna see another micrograph and guess which part of the body these bacteria are from. Can you tell us, Jenny, where is this one coming from?
Well, I can give a little bit of a hint too. So if you remember seeing a piece of what looked like wood here, so things that we eat have fibers, right?
Yeah.
Kind of look a little woody. So this is actually in the intestine.
Yes.
There's a lot of bacteria in your intestine. Bacteria help us break down the food that we eat and help us get out all the nutrients from it. We would honestly not really be able to survive without the bacteria in our gut and in our intestine, our microbiome.
And as you can see here, the predominant bacteria, the bacteria that you find the most, is this sort of like shorty, elongated bacteria. These are the typical form that all the coliforms, similar to Escherichia coli, or the lactobacteria, that is the bacteria also found in the yogurt, for example. So that's the majority of the bacteria in the gut that helps us having a healthy digestion and a healthy body, are these kind of bacteria. And the last one, but not least, this one, we're gonna see another micrograph and try to guess where this is from and maybe see if there's any difference with the other photos that you saw before. This one makes people laugh a lot when we tell them where it's coming from.
Yeah.
So can you tell us where it's coming from?
Well, first off, we could think that it looks pretty different, right? There's almost only round bacteria here, pretty circular bacteria. So it's not really inside us in the same way that the other pictures were. But if you can think about a spot on your body where maybe there's a collection of some stuff.
Maybe a spot that doesn't get cleaned very thoroughly, very often.
As often as it should. It's actually your belly button. So there's a little collection of bacteria in your belly button. There's actually bacteria all over our skin, and they also help us.
Yes.
So yes, when you cut yourself, maybe it's not great, but the bacteria in our skin also kind of protect us from worse bacteria that could make us sick. Many of these wouldn't make us sick
no,
at all.
actually, the bacteria that is in our skin and in our flora, either inside intestines, it's essential to have a proper immune system. It is so important that now it is considered an essential part of the innate immune system, which is everything that protects us from infections and from external agents like viruses or bacteria that can make us sick. As we were saying, bacteria are very, 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 very tiny. We need to have these micrographs and these microscopes to see them. But is that the only way that you can see bacteria?
No. So there's actually a few examples where we can see bacteria with our bare eyes, right? Pretty cool.
Yes.
We have an example here where actually even from a plane, you can see bacteria because there's so many of them at once.
Yeah.
So you don't see the individual ones, but you see the total.
Yes.
So here we have a picture of a lake
Yeah.
that's turned green from the amount of bacteria in them. We also show you a micrograph of the bacteria so that you can see how small they actually are. They're about the same size as the other bacteria we've shown.
So you have the difference between the individual single bacteria and then the community of the bacteria when there are many of them growing together. So knowing this, that you can actually see bacteria with your bare eyes if there are enough of them, now we can go ahead and we can test if there is bacteria in a lot of different places. Because we can sample those places, we can put them and give them some food so they can grow, and then we can see if there is bacteria growing on them or not by seeing if there are bacterial colonies or not.
And a bacterial colony is really like a pile of bacteria growing on itself.
It's like a big family all living together in the same place because they all come from one bacteria that started dividing and creating a lot of sister, daughters bacteria that we call it. So now that we know that we are actually able to see bacteria and to sample if there are bacteria in different places, I think it would be pretty fun if we check where can we find bacteria. And you actually took some samples, right, from some places?
I will see what I got.
Yes.
We already got some samples, and we're going to get some more in our lab itself.
We go to the lab and we take some other samples, and then we see you back for the results as well.
Yeah. This is a real-life used lab. A lot of research going on here in the background, but we're going to show you guys some more basic things about how bacteria work, where we can find them, what can kill them, and that sort of thing. But just so you know, there can be people passing by and things going on in the background and noises, and that's exactly how this place is. If we want to see the bacteria in the world around us, then we have to be able to grow them, because you can't see an individual bacteria, right? We have to see kind of the pile of bacteria. So we have a few samples here from some fun places. We have a soil sample, and we have a cat poop sample, my own cats. They were a bit skeptical as to why I was stealing their poop. So what we do is plate these samples on agar plates. So these are kind of squishy. They're kind of like jelly. So I'm going to go ahead and use these glass beads to spread the samples on the plates. So we're going to start with the soil. And then we're going to go ahead and plate the soil sample. And same thing with the cat poop. We give it a bit of a mix. And plate. All right, then we shake them. So we shake the plates to spread the bacteria across them. So then we just have to throw these beads away. But regular things around us that we touch every day, like keys, for example. Do we think there are bacteria there? Can be fun to find out if I can get my keys off my lanyard. So I'm going to try this one and just put it down on the plate. Now the tricky part is getting it back up again. Oh, first I'll push down to make sure it's there. Okay. What else do we want to try? We can try rings. So that can also be something that carries bacteria. Both of my rings. We'll see how good I am at washing my hands. People often forget to move rings around when they wash their hands. We can try a clean fork. See how clean it really is. So I'm going to go ahead and roll the fork on the plate. See how good people in this lab are at doing the dishes. And there's also a lot of bacteria in our hair. So I'm actually going to go ahead and test my hair tie as well. Try to get the hair off though first. But this isn't exactly something you clean that often, huh? So I'm going to try to twist it up so it doesn't go everywhere and ruin everything. Ooh, let's see if I can do this well. Okay. Here we go. So we looked at a few pictures of bacteria in our bodies, and we kind of want to try also to see, can we visualize this in plates in the same way? So what we're going to do is sample the bacteria in us. Ava, do you want to help me out?
Sure.
So we're going to start with tongues,
Oh.
I thought.
Okay.
Lick the plate. So these are clean plates. We know that. There's no bacteria on them. I'm not condoning people lick stuff randomly,
No.
but let's go for it.
All right.
Ready?
Yeah.
Nice and salty. Okay. So another thing I thought we would test is the ear. Because there's also a lot of hair around and everything. We can see if we see some fun patterns maybe.
Cool.
So do you want to go first or should I?
I can do yours.
Okay. Now you can
Jenny,
get a good
here.
grip on it. See if you see my earring in bacteria. Chili. And wet. All right, let's see yours. There we go. Oh, I hope I got enough. We'll see. I'm not sure we want to see the results of that. But we're supposed to have bacteria on it, so it's not, we're expecting there to be bacteria on us.
Yeah.
Another way we can test things is by swabbing a wet cotton swab on something and then rubbing it on the plate and seeing if things grow. So one of the things we're going to test is my phone. Again, phones are disgusting, so I'm not really looking forward to seeing this, but we'll see how it goes. I have my phone here. So I have to wet the tube first, or wet the cotton swab, I mean. And I'm going to just go ahead, swab all over the screen. And for good measure, I'm going to go ahead and swab this side too, which I'm sure I'm worse about cleaning. And then I'm going to go ahead and spread this on the plate. Okay. So we're going to do the same thing to some other surfaces around here that theoretically maybe should be cleaned but are touched very often. So we'll see how clean this lab is.
Ba-ba-da-ba-ba.
we're going to incubate these plates in these big incubators that look kind of like giant, I don't know, cabinets.
Room cabinets.
Yep. Go ahead and put them here. Nice and toasty. So now it's been a few days, and we've been able to incubate our plates. We started incubating them at 37 degrees, right? But some of these things we actually realized after a while actually grow better at room temperature. So if you think about it, I mean, 37 degrees is body temperature. So things in the body grow better at 37, but things on our skin or in the environment maybe grow better at 20 degrees. For most of these plates, we did starting at 37 for one night and then one night at room temperature.
What did you see, Jenny?
So we can start with our cat poop plate. Here we see, interestingly enough, my cats seem to only have one species of bacteria that they carry, but there's a lot of it, which is maybe not so surprising. We carry a lot of bacteria in our guts, and they generally, as we said, are good for us, right?
We saw in that micrograph the majority of the bacteria that we have in our guts tend to be like coliforms or lactobacteria that have similar shapes in the bacteria themselves, and also perhaps they make similar colony looking. So that's why it looks like it's almost all the same bacteria. It might be there are different species, but of course just looking at the colonies we cannot really tell.
Yeah, but we see a very different result when we look at the soil sample that we have. So here we see lots of different kinds of bacteria, and they all have kind of different shapes, the colonies too. So more different colors, different shapes, different styles.
Different sizes.
Different sizes, yeah. Some things grew much better than others.
And this is because in the soil, generally, there is a lot of different things, especially if you have a soil from a household where it's going to be more in contact with humans. It might also
And
have
cats.
and cats, of
In my
course.
case.
It might have some bacteria that are related to the human flora. Then,
Yeah.
of course, you are going to have not only bacteria, you are going to have other microorganisms, like could be some fungi, some molds. And actually, we can see here in the plate really well how we have a lot of different colonies. And I suspect that those bigger colonies that we have over here, those might be perhaps the fungi type of...
They kind of have fingers coming from the colonies.
which could be like the hyphae
Yeah.
from the fungi in the soil itself.
But it's hard to know what these are. So that's important to keep in mind when you're growing bacteria from an unknown sample, from especially soil, especially feces. You got to be very careful with it because you're growing large amounts of unknown bacteria. You have to treat them as worst case dangerous
Yes,
bacteria.
that's why we are not opening these
Yeah.
plates right now.
These we are extra careful with. Some other things that are things that us humans maybe touch more often. So we can start with our the plate that we did random stuff on,
Yeah.
kind of. So here we have, if you remember, we put my rings,
Yeah, we put
on
your
the plate.
rings,
my keys.
and your rings were
My rings were up here.
in this area
Yeah.
here.
And then the keys were here. Unfortunately, the plate got dropped later, so there's a crack in it, but that's not from anything we put on it. This is the fork. You
Fork,
can kind
yeah.
of maybe see the
Shape of
The
the,
shape
the
of the
three,
fork?
1, 2, 3,
prongs. That was a clean fork, remember?
supposedly.
And then you see the coil for my hair tie here.
This is the most striking one, and the one that you can see the shape of the thing
Exactly.
we sampled the most.
Yeah.
You can see perfectly that everything that tie touched, there is a colony growing.
Yeah, and some different colors here. There's a lot of diversity in how these colonies can look. So then we also have a plate with my daughter's hands and feet. This is a slightly concerning plate as a parent, but remember this is actually totally normal.
Yeah.
So the top of the plate here, you see my daughter's hand. So she kind of dug her hand into the agar of the plate. So the agar is the jelly
The
stuff.
food for the
food
bacteria.
For the bacteria, yeah. So you can see that she kind of dug her hand all the way through there, and there was bacteria in pretty much every single finger. You can kind of see the forms of her fingers there. And down here at the bottom, you can see kind of her foot. So this is the pad of her foot with her toes, and this is the heel.
I really like that you can almost see the shape of the toe. It's
Yeah.
so cute.
It is quite nice.
Yes.
Most people take, like plasters of their kids' feet, and I take
Bacterial sample.
samples. But yeah, so
That
not
means,
totally
yeah,
newly washed kid, but a relatively clean kid.
I mean, kids normally have their hands in their mouth all the time,
Yeah.
and mouth is a place where you have a lot of different bacteria and a lot
But
of
I also
it,
wouldn't
so-
expect anything different from us
No.
humans, us adults. It's just, in this case, her feet and hands fit better on a plate than ours do.
Exactly. Yeah.
But we did take some samples from us, remember?
Yes, we did try to see if we have a lot of bacteria on us or not.
So
And
let's
what
start
kind?
Let's start with our ears.
Yes.
We actually have quite different samples here. Let's start with Ava's. So Ava actually didn't have a lot of bacteria in her ears, or maybe I also didn't push hard enough. So a lot of this comes down to sampling technique also, if everybody does it the same way. But you see that there's a little bit there as well.
Some yellow bacteria, some white bacteria, which are typical skin bacteria that we can find.
And also, I mean, if you get hair touching as well, it's usually the same kind of bacteria in hair. Mine has a little bit of a crack in it as well, but it's quite a bit more and a little bit more diverse.
Yeah.
You do see actually a little bit of these red-orange colonies here
Yeah,
as well.
this is something that
Gonna
follows
come
you.
back a little bit, yeah. But a little bit more bacteria on mine. Then we also did our tongues, and this is pretty interesting. I don't even know if you're going to be able to see too much of Ava's, but Ava had an incredibly clean tongue.
There is a little bit of a background growth, very, very small colonies. I would suspect that those are very slow growing because you had them 2 nights incubating. And then there are some bigger colonies, normal size colonies for the amount of incubation, which, it means like
Ava's got a clean tongue. You clean your teeth.
Much bacteria in my mouth, even though it's completely normal to have a lot of different bacteria, especially in the teeth and everything.
Yeah. So I have a bit more. You can see mine as well here. You see this background again, the kind of slow-growing bacteria growing in the background, but I do have more of the bigger bacteria there as well. But we did take some swab samples too, which I find really interesting. So usually we don't really do this when we do these samples because it's not so interesting to show somebody this is what we swabbed. It's nicer to see like the form.
But now you just saw where we actually took the samples from, and now we have the results here.
So we can start with, this was actually my phone. So you see these kind of the red and the yellow bacteria coming back. And again, here we see quite a big diversity.
Yes.
And I have to say, usually I sanitize my phone because I work in a lab. So I can get bacteria on my phone. So I sanitize it regularly. Here I intentionally didn't sanitize it for only, I think, 3 days. So this was 3 days of stuff from a phone. Phones are pretty gross when you think about it. This was our lab's coffee machine.
And this is actually super interesting.
Yeah.
Because
We have a clear mold.
obviously the coffee machine gets cleaned by itself with this automatic system, but the surrounding areas not so much, and it's an area that it's generally very humid all the time. And this is the only sample that we actually got this clear mold growing
Yeah,
in
a
it.
very obvious mold.
So this is the typical mold that you can find, in bread probably.
I would say this is probably what most people are used to seeing.
Yes.
Yeah.
Yes. So this very distinctive from the specific environment we took the sample, and also some sort of bacteria, but not so many. So it wasn't
Not
that.
as bad as I thought it could
Yeah,
be.
exactly.
However, our lab computer's keyboard, the computer keyboard, also, I mean, a lot of skin bacteria, what we saw before.
Yeah, the typical yellow bacteria that you might get in this skin, or like skin samples or things that skin has touched, they tend to be Staphylococcus aureus bacteria. That's
Or
where
other
the names come
Staphylococcus,
from, or other
yeah.
Staphylococcus. What I think is interesting is if you get your plate from your phone,
Yes.
and put it next to it, because of course the phone plate is sampling the skin flora. If you see, this would be the flora that is associated with Jenny in particular because it's her phone, but this one is more a representative of the whole lab because everybody comes to this computer and touch the keyboard.
So
you
it's all
can
my
see
colleagues
that it
as well.
depends from person to person. The diversity and the flora in the skin can vary a lot, so much that you can even see it just with this kind of easy sampling in plates.
My phone sample actually looks closer to my ear sample, more than the keyboard, even though I don't touch my ears that often.
But if it's your skin and that's
Exactly, it's
your
the
own
skin,
flora,
either
and
place.
it's something that is really important inherently to your own health and to your own body.
Yeah, it's important to say that most of these bacteria, I mean, obviously we walk around with bacteria on us all the time. We usually don't get sick. I mean, you wash your hands before cooking, especially if you're cooking certain things. And especially right now in these times, we wash our hands maybe a lot more often. But I mean, this is normal and this is natural and not unhealthy.
Yeah.
However, this is a place that you want to have a bit clean. This was the toilet seat that we sampled. And it is quite clean.
Pretty clean,
Yeah,
yes.
which is not that surprising when you think about it. I mean, you clean your toilet more often than you maybe clean your phone.
On your keyboard, obviously, as I see. So,
Yeah.
so
that's comforting.
Yeah, it is indeed.
And a door handle was also pretty clean. Not too much growing there. That's also kind of, I mean, it's something that you touch, but it's
Something
kinda
that gets
gets
cleaned
brushed
often.
off
Yeah.
and cleaned.
It's
Yeah.
part of the routinary cleaning, you know, like when
Yeah.
you clean surfaces, you always clean doorknobs, but you don't clean perhaps phones so often.
No. That's what we've seen on these environmental plates that we've taken. We see a bit of diversity, and like we said, some expected, some unexpected things, and I hope you guys learned a lot from
Yeah,
that.
I hope you like seeing these different colonies and the plates. I think they're very exciting, and they teach us a lot. We can
Yeah.
actually understand a lot of the things that are around us.
That
when
we can't
look
really
at this.
see
Yeah,
usually.
it's really cool.
So now that we've seen where bacteria can be, and we've seen that even on clean objects that we know are safe, they can be pretty much everywhere, right? And a lot of them, especially on us, in us, everything like that. So we know that bacteria around us aren't necessarily bad, right? We're not sick all the time.
No.
Things are pretty good.
And the majority of the bacteria are actually either harmless or beneficial.
Exactly, yeah. But sometimes you do get sick from bacteria. So a lot of people may have had a strep throat sometime in their life or other infections. Stomach bugs can be caused by bacteria.
Some urinary tract infections
Urinary
as well.
tract infections,
pretty
yeah.
common.
So we do occasionally need to kill bacteria. And it can be good to know how to.
Yes.
So if you're trying to kill bacteria and you've never, we didn't have antibiotics before, you didn't know where anything comes from, you're kind of just trying random things. What's something you might try to kill bacteria?
What's something I might try? Something that is strong, I would say, right?
And oftentimes these old remedies that we have that are passed down from in family, they're often associated with food, right?
Yeah, some natural.
Natural products, food, and that sort of thing. So maybe there's some food that kills bacteria
Yes.
itself.
So I think it should be fun to go ahead and maybe test some strong foods, like it could be garlic, like it could be chili peppers,
Yeah.
that ginger, for example. I think people.
Yeah, I think a lot of people have heard of ginger to treat a sore throat, maybe.
Yes. So I think it would be fun to go back to the lab and then put these foods into plates with bacteria and then see if they are able to kill bacteria or not.
Sounds good.
Let's do it.
So what I'm going to do is first spread some bacteria on some plates. So we're going to use cotton swabs that are sterile. I have some bacteria that I need to mix. Then I just dip the swab in the mix and spread it on the plate. So this way, what we're going to see after the bacteria have grown is bacteria covering every part of the plate. Which means when we put something on the plate, on top of the bacteria, we can see where they don't grow, see if they die. Take a fresh one of these. So this is just regular lab bacteria. They're very, very safe. I usually tell people that you can theoretically drink a glass of this and nothing will happen to you bad. So then I have some foods that we want to try. We're going to start with ginger. So I'm going to try to scoop up some of this ginger, get some of the nice juicy gingerness of it to put on the plate. This is easier said than done. Try to spread some of this stuff out. I have a bunch of different kinds of chili, so I'm excited to see what happens with this one. There we go. And same thing here, we want to get a lot of the chili juice to see that might be where the real power lies. Like that. And then the habanero. Smells pretty strong in here now, huh? Okay, so we've got a nice colorful chili plate. Historically, one of the places where we've found natural antibiotics is actually in molds. So think, moldy bread, moldy vegetables, moldy fruits. We're not sure about this one. I've never tried this one personally, but I found a moldy kiwi, so I'm going to see if this one works. Now this doesn't smell quite as good. I think one's enough. We have some garlic. Smells really strong. Maybe it'll work. We'll see. In this case, I want to get some of the nice garlic juice up as well. Oh, that's a strong smell.
You can check Philip.
Okay. And we all know lemon is sour, so the idea might be that this might also kill bacteria. I have a slice of lemon. I'm just going to put that straight on the plate, and we're going to see if this has any effect on the bacteria. Press out a little bit of the juice from it, so we see if it works. There we go.
After having incubated the plates that contain simple lab bacteria with some foods that we want to check if they actually can kill bacteria or not, let's start with one that, of course, a lot of people think that might kill bacteria, and because it's quite strong, because it repulses a lot of people also, like how strong it is. And bacteria seem to not like it at all.
No.
And this is the garlic sample. As you guys can see here, there is a pretty intense zone of inhibition. So the way that we are checking these plates is everything that you see cloudy like this is the bacteria that we put in the sample. It was of course transparent when we were actually sampling it because it was really small, but after one night growing, it becomes like this. In the middle we put the food that we want to test, and what we see is that there is no bacteria growing at all for really big area around the food in itself. This means that whatever substance was in the garlic diffused through the agar, and then the bacteria was like, nope, when this thing is here, I don't want to grow.
Not interested.
Not interested in growing, or even cleaning it actively. It depends. We cannot really say which one of the two options it is. But as you can see, garlic, it's pretty strong antibacterial, and this we have known for long time, I would say, in human history, that garlic somehow can do something towards microbes. The thing is that the compound that does this to the bacteria, it gets completely inactivated when it gets to the stomach.
Yeah,
so of course,
your stomach acid kills it.
eating a ton of garlic is not going to help you get rid of an infection in some part of your body. In principle, if you would cover all surfaces with garlic juice, bathe in garlic, you could potentially, get rid of a lot of bacteria, but that's not really the point. No. We just wanted to mention that.
You got to question if it's worth it at that point. Do you want to smell like garlic all the time very strongly?
What we can see is that, there are natural things in food products or in some natural that can actually kill bacteria. Another plate that is perhaps a little bit surprising, because I would expect that the strong chili peppers might actually kill bacteria.
You'd think. I mean, you think of the spicy everything, like it should be killing stuff.
But what we can see here is actually not so much effect when it comes to the inhibition. At least, of course, not as strong as the garlic, definitely. No. We can maybe see some sort of like effect very close to the green chili, but not so much or almost nothing into the red chili and the yellow chili.
And most of what you see, it's not necessarily even that the bacteria are dying or not being able to grow at all. It's that you see that they're growing slower. So we don't see as much bacteria around the chili while we still...
It smells tasty.
It smells so strong, this one actually did smell quite nice. But we do see that the bacteria aren't growing as fast around the chili. So they are affected. Yeah. They're not killed. They're not completely stopped from growing.
One that is actually pretty interesting. And
this is new for us.
We're trying, we had never really tasted the kiwi that was a little bit moldy that you sampled. And we are a little bit surprised by the results because we do see an effect, quite a strong effect around it. Of course, not as much clear as the garlic, but still there is an effect around. But the area where there is more mold growing, which will be up here, as you can see.
You can even see the mold still growing on the kiwi, so the mold has continued growing on the kiwi.
In that part, there doesn't seem to be a stronger effect of the killing or the inhibition. So what we're thinking is happening around here is that the acidity of the kiwi is being spread through the agar, and is this acidic condition, this acidic environment that is not allowing the bacteria to grow. And we have a control sample for this acidity, which is we also plated a lemon. And as you can see, the lemon effect is really strong, but we do know that lemon doesn't have any any component that is actively antibacterial.
Yeah, it's the acidity of the lemon.
We know that the juice is very acidic. The acidity kind of expressed through the agar, and then the bacteria don't like growing.
Yeah.
These particular bacteria don't like growing in this acidic condition. There are some bacteria that love acidity, but they are actually not this kind of bacteria.
And I mean, this comes kind of naturally. So if you think about it, if you cut your finger and you squeeze a lemon, it's so painful, right?
It is, yeah.
Yeah, so it makes a bit of sense that this would be.
Yeah. So you can see very,
bacteria.
very clear, and if we compare perhaps the kiwi and the lemon, you see that it looks a little bit similar.
Yeah.
So the kiwi effect might not be the direct mold that we were trying to
Yeah, it's hard to
do.
know if
No.
and it might also be that the mold is preventing the growth a little bit, but not killing it, and the killing, or like the stronger effect is the acidity. It's hard to tell.
Yes. And we are going to end up with a little bit of a controversial food item that we
Yes.
test here in this sampling, and it's the ginger. And this comes with a disclaimer. We know that a lot of people think that ginger helps somehow when you are feeling down with a cold, which might or not be bacterial or viral related.
Usually viral.
Usually viral. And a lot of people make like ginger teas, and they do feel better. And we don't want to make you think that you are not right and that you don't feel better when you're taking these teas, because they certainly help. But when it comes to testing if they can kill bacteria, what we see is that there is no effect whatsoever.
Yeah.
So they cannot kill bacteria. Ginger per se cannot kill bacteria, but
It can certainly make you feel better. It can be anti-inflammatory. It might, things don't swell up as much. The effects from the bacteria might not feel as strong.
it's also an immunobooster, so it
Exactly.
has been shown to help the immune system to get rid of whatever infection is
Yeah.
happening.
But the ginger itself is not killing any bacteria.
No, and this you can see very clearly in this sample, especially if we take one of the things that can kill bacteria.
So let's go back to the garlic and compare the two.
Yeah, so we can compare
Keeping in mind that garlic doesn't kill bacteria in you either, but
outside, which is the sample, the conditions that we're testing it, you can see that there is no effect from the ginger. Although ginger is very tasty and is present in a lot of tasty food.
as I say, all these things combined minus the moldy chili makes a pretty delicious dinner. Also
Yes.
minus the bacteria.
It's really good.
Yeah.
So as you can see, there is a way that we can test if some foods might have antibacterial properties and kill bacteria in the lab conditions and in the situation that we're testing them in. And we got some surprising results, some things that maybe...
They're very easy to see too.
Yes,
Very
exactly.
visual.
And it's also kind of fun to test.
Yeah.
Cool.
And it smells nice.
We've seen what food works, what food doesn't work, and what I want you to know is that actually antibiotics or something that could kill bacteria was firstly found in also a natural product. This is probably you have heard of Alexander Fleming. Alexander Fleming was a not so clean lab technician back in
Yeah.
the days. In the
Most labs weren't back in the day.
Yes. So he had some plates of the kind that you have seen in the video thus far, and he had some plates. He went on to holidays, and when he came back, he saw that some of the plates had grown so old that they had mold growing in them. The same mold that you can find if you leave your oranges for way too long at home, or perhaps lemons, you forget a lemon in the corner of the fridge.
Or in his case, it was actually moldy bread.
Or moldy bread as well, yeah. There's different kind of molds that grow on foods. And what he noticed, he was smart enough to notice that around the mold growing in these plates, the bacteria that was supposed to be there, because he was working with bacteria, it was completely gone. And he was like, maybe this has something that can actually kill bacteria. And this antibiotic is the one that we know as penicillin, because it actually comes from the mold Penicillium.
And actually, I mean, in Sweden, a lot of people use the phrase penicillin and antibiotic interchangeably. They call all antibiotics penicillin.
Interesting.
Because this is the first and often a still commonly used one, not exactly the same, but a derivative of that one is still commonly used.
And talking about derivatives, we have seen that some food actually have compounds that can kill bacteria. But if we get sick and we go to the doctor and the doctor really recommends that we have to take antibiotics, it's not enough with just eating a lot of this food that might have some
No.
antibiotic properties.
You can't
So-
bathe in garlic, can you? Or I wouldn't suggest it. Something might kill bacteria in a dish, but is it ever going to work in us? It has to be functional. It has to get to the right spot. It gets pretty complicated pretty fast. So what we actually give patients who are sick is what's called antibiotics.
Yes.
That are often maybe from a natural source, but they're produced not naturally. You don't give them the food itself.
Because you need to have the right concentration, and you also need to make the antibiotic be able to get to the place of the infection. So for that, a lot of scientists for many years have done hard work to find the ways that these drugs can work as a medicine.
Changing little details at a time usually until we find something that works as good as we can make it work.
But what we can actually do in the lab is to test if a bacteria can be killed or cannot be killed by a certain antibiotic. And this is the test that is normally done in the labs, in the clinics, and in the hospitals when a patient has a bacterial infection and they want to test if they can use an antibiotic or they cannot use it. They use the same technique that we are going to go back to the lab and try to show you how we can test if we can use an antibiotic or not against a specific bacteria. Do you want to go to the lab?
Sounds good.
Let's go.
So we're going to test if certain antibiotics can kill the bacteria that we're looking at. In order to do that, we have these little paper discs that are kind of drenched in antibiotics. So when we put the little paper pieces on the plates with the bacteria on them, the antibiotics spread out into the plate. And hopefully, they can kill the bacteria that we're looking at. Because that's what we want. We want to be able to kill bacteria when they're causing an infection or causing us harm. So first I need to spread some bacteria on the plates. I have 2 different kinds of bacteria that I'm going to spread. And I'm going to do this the same way I did before. So these are really 2 very similar bacteria strains, but one of them has gained some genes. So we can see if those genes impact whether or not the antibiotics are able to work against the bacteria. So then I'm going to try to put these discs on the plate. Sometimes it gets a little bit messy, so we'll see how nicely this looks, but I have 3 different kinds of antibiotic discs here, and I want to get one of each on the plates. I use this little needle to help me. And just kind of poke it on. There's one of those. So then I've got one of each kind of antibiotic on this plate. Do the same for the other plate. Okay. So then after the bacteria have grown, or not grown close to the antibiotic disc, we should be able to see how well that antibiotic works against the bacteria.
If you remember, what we did was to plate different bacteria that we know are more sensitive to some antibiotics than others, and we put also different antibiotics so we can test different antibiotics at the same time. So here what you can see is that this particular bacteria that we put in this plate has 3 different antibiotics, and there is a big zone of inhibition around 2 of those antibiotics, but there is no inhibition at all around this 3rd antibiotic. That means the bacteria here doesn't really mind whatever antibiotic was in this disc, whereas the antibiotics that are in these 2 discs are effective. They work against this particular bacteria. Here, a lot of inhibition, here, a lot of inhibition. We don't really make differences, or we don't make assessment in how big these circles are so far. It's just that we see they work. This bacteria is sensitive to these particular 2 antibiotics. On the other side, when we have this other plate, what we can see is that this one is sensitive to only one of the antibiotics, whereas it's resistant to the other 2 antibiotics. If we put them next to each other, what we can see is that both bacteria are sensitive to this particular antibiotic here, but this bacteria has become resistant to this antibiotic because here you have the zone of inhibition. Here this antibiotic is working, but here it's completely overgrown with bacteria around it, so it has become resistant to this particular antibiotic.
And it's worth keeping in mind, these are actually very, very, very similar bacteria. It's the same species, or the same strain even, that's just gotten a few extra genes through something called a plasmid. So it's by that way become resistant to the antibiotic.
Yeah, so we can actually see these 2 bacteria are related, they're very similar, but something has happened that now it makes it able to grow with the presence of this particular antibiotic, where before it would be completely killed. And this is what is called the acquisition of antibiotic resistance, in particular through maybe horizontal gene transfer, which is the transfer of these particular plasmids, which are extra material
outside of this.
Outside of the normal DNA of the bacteria, they acquire something, and then they have like a superpower, like a little bit like this one, gets like a super cape. It acquires a resistance to a specific antibiotic.
Yeah, which is what happened in this case. It's the addition of new genes. So now we've seen how this ability of antibiotics to kill bacteria can be seen in the lab. We use these little discs, and we can measure the diameter of the zone that's clear, the zone of inhibition, as we call it. But the reason why these antibiotics maybe don't work against the bacteria can differ a lot. So some bacteria, just by how they are, I mean, they're the way they are naturally, the antibiotics won't work against that specific kind of bacteria. So like if we look at this bacteria behind Ava here, it has a pretty thick wall here, right? And depending on how this wall is built, maybe the antibiotics just can't pass it. In this case, it doesn't mean that there's anything that's changed about the bacteria themselves. It's just that that antibiotic doesn't work against that bacteria, which is why we often need to know what bacteria is causing what infection. It helps the doctor a lot to know how to treat that. But other times, we maybe have antibiotics that we think should work against this bacteria, but they don't. This bacteria has become resistant instead. And if you see in here, this purple stuff, this is this bacterium's genetic material. And bacteria can actually develop the ability to resist antibiotics. They maybe break them down sometimes. So something in here is coding for something that breaks down antibiotics, or maybe builds in a little pump that pumps out the antibiotics from the cell.
So the antibiotic comes in, but before it can kill the bacteria, it's pumped out again.
Yeah. Or if the antibiotics are targeting something in the cell, maybe a mutation occurs and that target in the cell is changed and it no longer really works against the bacteria. So this is a constant thing. I mean, bacteria are always changing. They're always evolving. There's natural mutations occurring, that sort of thing. It's nothing that's really surprising to us. We kind of expect it to happen.
Yes.
The problem is that it's happening so much now.
And it's happening really fast because we're using a lot of antibiotics, and sometimes we're using them for things that are not supposed to be used for.
Yeah.
Perhaps you have heard about the use of antibiotic in animal production, because it's easier to give antibiotics to the animals than having to keep them in conditions that will reduce the possibilities of getting infections. So a lot of people used to just give them antibiotics to prevent the infections in the animal husbandry. Also,
sometimes a viral infection looks like something that might be caused by bacteria. It's if the doctor doesn't know the difference between these, I mean, it's hard to tell. It's not that the doctors aren't doing their best. It's that it's hard to tell, and sometimes you need to treat somebody pretty fast. And we have a few examples here I kind of want to show real quick.
yes.
Not all viruses look like this, but we have two examples of viruses that look about like this. Well, if we compare to some bacteria examples we have, or the bacteria that we have here, they look a bit different. But it's also worth bringing up, these are much smaller.
Much smaller. And actually, I think it might be even up to scale. This one here is a virus that actually infects bacteria, not human cells or animal cells. And this is actually kind of like maybe a little bit up to scale. Maybe this one should be even smaller.
As I say, I think it should be even smaller.
Even smaller. Because this one will come here and it will try to go into the bacterial cell the same way that another virus goes into our cells, if it's a human virus. So we want you to really see that there is a lot of difference in how they look, but also the
how they work and how they
sizes of it as well.
Yeah, how they do things.
It's very important that we think about these things, that antibiotics are not just expendable medicines that we can use at any time, in any moment, and for anything. We should use them only when a bacterial infection is suspected and confirmed, and if your doctor tells you that your bacterial infection is not strong enough or bad enough or the kind that will go away by itself, that antibiotics are not needed, that we don't really think we should have them because they exist, because we have to say them.
Yeah. And on that note, you mentioned before that antibiotics have actually changed how medicine looks today. So, Ava, do you know some medical advances that have only been possible due to antibiotics?
Yes, it's very important to think that, for example, things that we think are common and that we cannot even imagine medicine without them. For example, surgeries that extend the quality of life of our elderly, like it could be hip replacements. Those surgeries are not life or death surgery. They are just increasing the quality of the life, but they are risky because any type of surgery, anything that is opening a body and changing parts of the body has the risk of infections. So we need to have antibiotics that work in case these patients get infections. The same with with patients that are on particular risk of infections, like could be immunodepressed patients, like patients that have gone through transplants, or patients that are undergoing chemotherapy because their own immune system is not working properly, so they are more at risk of getting an infection from normal bacteria that normally don't get any harm to a healthy individual. Those are very important to have antibiotics as well.
Absolutely. And there's a lot of common medications nowadays that actually decrease our immune system, because oftentimes a disease can be caused by an overactive immune system. It's not always only for good. So it's important that we keep these things in check, that we use antibiotics only when necessary.
C-sections, for example, are also very rely a lot in antibiotics, and some of the bigger measures that they were talking about in case resistance gets really, really bad is to stop doing these kind of surgeries, because a planned C-section might not be a life or death situation. So they're only going to do it if they really, really need to, because they don't want to put the patients at risk of an infection that cannot be cured. But the whole thing is not as grim as we're putting it here, because we are actually doing something about it. And because we're here talking to you about bacteria and antibiotics and antibiotic resistance, we think that increasing the overall knowledge of this topic is going to be the way to reduce the chances that resistance gets so bad that we cannot do these surgeries anymore, and that we are not going to be able to treat a regular infection like we are today.
So it is important to think about, okay, what can we do to prevent the use of antibiotics? As an individual, you can think about where your food comes from and how it's made. That plays a pretty big role. You can think about preventing infections. So if we help many parts of the world get clean water and all these other things. Proper sanitation. Less infections equals less antibiotics used. Good for everybody.
Proper diagnostics. So if we have technologies that are advanced enough to give us an answer if an infection is either bacterial or viral, and within bacterial, what kind of bacteria it is, if we can have this quickly, we're going to be able to use antibiotics much more selectively and better. Yeah, so as you can see here, there are a lot of aspects to the issue of antibiotic resistance, but what you can do as an individual in your day-to-day basis is try to prevent the spread of bacteria as much as possible. And also, if your doctor is telling you you are not needed antibiotic treatment, trust your doctor, because that would be the best, both for you and for the common good of the antibiotics for everyone. And please, if the doctor says that you have to take antibiotics, I know that as soon as you started taking them, you're going to feel much, much better, but you have to follow the instructions and finish the whole course of antibiotics in order to also help out the spread of resistance or the accumulation of resistance in a sense.
So Eva, if people have been watching this video with us for a while, what are some central things we want them to remember?
So if you doze off a little bit watching this video, just pay attention now. We wanted you to know that bacteria are everywhere, that bacteria are majority harmless, a lot of them are actually beneficial for our health and our immune system, that they are really, really, really small, and you cannot see them with your bare eye individually. You need a microscope for that, but you can actually see them with your bare eyes if you can grow the bacteria and get bacterial colonies, and then you can go out there and test where bacteria might be found. We also learned that antibiotics, which are medicines that can kill bacteria, originally come from natural products, and still some food might have some antibacterial properties, but we have to have proper antibiotics as medicines in order to treat bacterial infections. And we are able to also test in the lab if these medicines are working or not working. When antibiotics stop working, what we have is a case of antibiotic resistance. And this makes the bacteria survive an antibiotic treatment. This is quite dangerous because that will mean that we cannot treat bacterial infections in people that get really sick. And it also might mean that the medicine as we know it today with transplants, C-sections, cancer therapy, hip replacements, for example, might stop looking like we know it today. And therefore, we need to make sure that we only use antibiotics when we really, really need it. I know we said this a lot, but this is very important. Just use antibiotics when they're needed.
Good finish there.
Yes. Thank you so much for being with us. We hope you enjoy this video and that you learn to love bacteria as much as we do, right?
Yeah.
Bye.
A little bit obsessed.